So welcome to my presentation, which is titled From Sewage to Safety, Digital PCR's Role in Tracking Microbial Threats to Support Public Health. My name is Gabriela Eder, and I'm a PhD student of Julia Vierheilig's working group at the Institute of Water Quality and Resource Management at TU Wien. Okay, after a short stop at the disclaimer slide, we jump right into the topic and have a look at the outline. So at the beginning, I would like to talk about how wastewater can act as a source of information. And then we will have a look at the research topics that we're working on. And with that, at the end, you will know how dPCR is used on a daily basis in our laboratory. So the two main research focuses we have is first of all wastewater-based epidemiology, where we monitor different pathogenic viruses, and on the other hand, is monitoring antibiotic-resistant genes, or short ARGs. And there we try to establish methods for upstream and downstream analysis. Okay, so how can wastewater act as a source of information? We have this little wastewater treatment plant in the middle of our slide, and then there are now two ways of tackling things. We can either look upstream and analyze the inflow, which would be wastewater-based epidemiology, or we can also check downstream and see what's going out and analyze the outflow, and this would be more like a water quality analysis. If we do the wastewater-based epidemiology, we can monitor the health status of the population that is connected to the wastewater treatment plant. And with this, we can also derive possible measures to protect public health. If we look at the water quality analysis on the right-hand side, we can monitor the health status of the environment. So we can see what is going back to the rivers. And with this, we can deduce potential hazards which might be arising from water use, like drinking or bathing. And then we can derive possible measures to ensure safe water use. And all of this together should serve the One Health approach, because the One Health approach has the idea to tightly connect human health with environmental health and animal health and see a more holistic picture rather than seeing the disciplines isolated from each other, because the common goal would be to protect public health. All right. So let's have a look at the two research focuses we have in our lab. First of all, the wastewater-based epidemiology. And there we analyze different viruses, could be either respiratory viruses like SARS-CoV-2, influenza, or RSV, but could also be other viruses like measles. And there we have a main project going on. It's called CSI Wastewater, and this is the project that I would like to introduce to you today. The second research focus we have is the analysis of different antibiotic-resistant genes, short ARGs, as already mentioned, like might be SAL1, INTL1, ERMB, TETW, or MCR1. There are so many different. And there are also some projects we're working on. One is called ARISE, and again, there's the CSI Wastewater project. But this time I will not talk about certain project. I would rather give you a general overview about what kind of research we do with antibiotic-resistant genes. But let's get started with the first topic, the wastewater-based epidemiology. So why wastewater-based epidemiology? There are many advantages. One would be, for instance, that the wastewater in general is the mirror of the society. So there's nothing really we can hide from the wastewater, no matter if it's different kind of chemicals or pathogens that are in the society, they will all end up in the wastewater. Then we have an early detection system. So we pretty much get the results in time, because when we get the sample in the morning, we basically have the results in the evening. Wastewater-based epidemiology is also quite cost-effective, especially if we compare it to testing different individuals, which of course would be way more effort to test thousands of people, which of course is still very helpful, but might cost a little bit more. And then we have less ethical concerns, because usually the wastewater results can never be traced back to any individuals. And then we have a high coverage. So an entire population group becomes a study participant without having to do anything else than what they would do anyways, and this is using the toilet. But of course, no advantages without disadvantages. There are always some limits as well. So one limit would be that we cannot detect a total number of infected people. It gives us more estimations. So are trends going up, are trends going down? It gives us a bigger picture. And then, of course, the wastewater composition may alter the signal. It could always happen that something ends up in the wastewater that maybe shouldn't end up there, or we are not aware of, and this might alter the signal. And additionally, there is no globally standardized method yet. However, there are people working on this. All right, so let's have a look at the CSI wastewater project. And this project basically started quite at the beginning of the SARS-CoV-2 pandemic when it reached Austria in early 2020. And the aim of this project was, and still is, to monitor Vienna's wastewater for pathogenic viruses as a source of information for the health authorities. The strategy is to sample the inflow of the Vienna main sewage treatment plant once up to several times a week. That's just depending on the severeness of the situation. And with this, we cover the entire population, the entire Viennese population. What we can also do is demand-oriented sampling of the 4 individual main sewers. So if you look at the map on the right-hand side, you can see the wastewater treatment plant and also the 4 different colors like green, yellow, red, and blue. And these would be the 4 different main sewer systems that we are able to also sample individually, which was especially important back then when numbers were really, really high, infection numbers, to have a closer look and get results in a higher resolution. And then, of course, there's the sample processing and communication part, which we have a closer look at now. So the methodology of the CSI wastewater project looks like this. So on the left-hand side, you see the city of Vienna, and then you see the toilets. And some of the people might be infected with different viruses. And then the wastewater comes to the wastewater treatment plant, and there we take a sample. And this starts the whole sampling process. We get a 24-hour composite sample, which includes the different viruses. And with the good thing about this is that we don't only just cover the entire city, we only cover an entire day. And this is why we do this 24-hour composite sampling. After that, we enrich the virus. We use a pack precipitation method, which is a very commonly used method for virus enrichment, and it also includes very extensive centrifugation steps. Once we have our enriched sample, which holds all the viruses, we try to get out of the viruses what we're actually interested in, and this would be the DNA and the RNA. So the next step would be the nucleic acid extraction, which we do with an automated device. And after that, we get our TNA extract, so total nucleic acid extract, which contains all the RNA and DNA. And after that, we can do the quantification. And since all of the viruses we're tracking on a regular basis are RNA viruses, we do reverse transcriptase, dPCR. And for all the viruses, which are here on the list, like SARS-CoV-2, influenza, RSV, and measles, we use the digital PCR. However, back then when we started, we only had a qPCR. So at the beginning, we only did qPCR measurements. And then when we got the device in the dPCR device in 2022, we started to do parallel measurements for quite a while. And then now in July last year, we decided to totally switch over to the digital PCR device. Once we have the results, we can of course have a look at the data and do the data analysis, and then we do the last thing, which is maybe the most important one, and that's communicating the results. We have weekly meetings with our project partners, we have weekly reports, and we also have a weekly update of our dashboard. Okay, so how does the current situation look like? On the left-hand side, you see a graph of the wastewater signal versus the inpatient admission to hospital. So what we like to do is that we compare our data that we measure the signal that we measure in the wastewater to publicly available data, like the bed occupancies of the hospital, to see if what we're measuring really makes sense. And luckily, we can see that the trends of the wastewater, which would be the little brown dots, and the bars in the back, so the bed occupancies, fit together quite nicely. On this graph, you can also see at which point, again, we started doing the dPCR measurements. Before that, as mentioned before, we did qPCR quantification. Additionally, I would like to show you a screenshot of our dashboard, which shows the wastewater signal throughout different seasons. And they can also see how the signal changed throughout the season, so that it was never really the same throughout the different years. And in the center of the graph, there are the winter months, because usually these are the months where people are more likely to get respiratory virus infections. But what you also can see is that right now, and the numbers are quite low, and we don't detect that much SARS-CoV-2 in the wastewater, which is, of course, what we like to see. The same thing is done for influenza and also for RSV, where in the top, again, you can see the wastewater compared to the bed occupancies of the hospitals. And again, you can see that these trends fit together quite nicely. And in the bottom, again, there's a screenshot of the dashboard. However, this dashboard is publicly available. So if you're interested, of course, you can scan the QR codes and have a look at it, because this is, as already mentioned, updated on a weekly basis. All right, so let's have a look at the second research topic we're working on, the antibiotic resistant genes. And there again, the question, why AMR monitoring? So first of all, AMR monitoring is antimicrobial resistance monitoring, which does not only cover bacteria, but also different kind of microorganisms, or all kind of microorganisms that can have resistances. And AMR is one of the top 10 threats to public health, according to the World Health Organization. And worldwide, there was nearly 1.3 deaths only in 2019, which were either attributed or associated to AMR resistances, according to Murray et al. And if you look at this graph on the right-hand side, you can see that especially in the sub-Saharan African region, people have basically the highest risk of getting infected with antimicrobial resistances, basically bacteria which are resistant, or other microorganisms. If we have a look at Europe, things aren't that much better. In the year 2020, there were more than 35,000 deaths that were associated with AMRs. And just to put this a bit into perspective, this equals approximately the amount of deaths due to influenza, tuberculosis, and HIV altogether. And finally, the projections, if we have a look in the future, that also doesn't look that bright, because there is a prediction that in 2050, there is a rise to 10 million deaths due to AMRs. And this number would then even exceed the number of deaths that are associated to cancer. So I think it's quite clear that AMR monitoring is something that is really important. And we at our institute, since we are the Institute for Water Quality and Resource Management, have a look at the aquatic environments of the AMRs. So what are basically the central challenges of AMR detection? So first of all, AMR wastewater monitoring is still in its infancy. So there's not that much of knowledge we can look back to, decades and decades, unfortunately not. Additionally, there is a very large number of different antibiotic resistances, thousands of different genes and gene variants. And there are many different diagnostic approaches out there. Another challenge is that the surveillance of AMR in wastewater became compulsory according to the new Urban Wastewater Treatment Directive, which was published last year. This is a challenge, of course, but on the other hand, that makes us happy, because now it is compulsory and it has to be tracked, which is important. And what are the goals now? So the goals would be to evaluate the different analysis methods for various objectives of surveillance. So either if it's the wastewater-based side, so upstream, or the water quality analysis side would be downstream. Then methods should be harmonized. Another goal would be a comprehensive determination of the status quo. And then it would be another goal to investigate temporal trends and to be able to detect emerging AMRs early in the community. And then the evaluation of the environment's role of the transmission of AMRs, and finally to conduct risk or exposure assessments of human but also animal health. Okay, so what are the analysis methods that we are currently using in the lab, or that are usually used for AMR detection? That would be either the cultivation-based methods or the molecular biological methods. The cultivation-based methods is just a direct cultivation of bacteria isolates. For instance, on this graph you can see there, or on this picture you can see like a plate with a dense bacterial lawn, and then we have the antibiotic platelets that are put on top. And then based on the inhibition area, the bacteria can be classified into either resistant, as it is the case in the middle dot, like the antibiotic platelet in the middle, because here the bacteria doesn't care if there's any antibiotic, or if it's intermediate, or if it's sensitive, as it would be the case in the left-hand side, because they cannot grow due to the antibiotic. The molecular biology methods are based on the detection of antibiotic-resistant genes, and that can be done very classical via dPCR or qPCR. So that's a targeted detection of selected resistant genes, and it gives us quantitative information. Another thing, however, that can be done is sequencing. This is non-targeted. So this is basically a screening of the entire genetic material of the sample to detect resistance genes and also their origin. This method is, however, limited. It gives us limited quantitative information. All right. So with this, we're nearly at the end of the presentation, and I would like to conclude what we've been talking about now. So biological threat monitoring enables a better risk assessment, and it paves the way for targeted actions by health authorities. So once they know, they can act. This, of course, requires sensitive, comparable, standardizable, but also robust methods. And PCR-based approaches, but also appropriate concentration and subsequent extraction methods are a promising method, or are promising methods to detect and quantify pathogenic viruses, but also antibiotic-resistant genes. And the monitoring of the different sectors, whether if it's the population upstream or the environment downstream, and also different targets, whether it's viruses or antibiotic-resistant genes, allows us to get a more holistic picture, and this, of course, serves the One Health approach. With this, I'm at the end of my presentation. I would like to thank all the project partners and, of course, the wonderful team at the Institute that I have the chance to work with. And thanks all of you for your attention.